All right, I think we can get started. Well, welcome everyone to the next topic in our Refsum webinar series, the science behind Refsum disease. I'm Christy DeMarco, and I'm the founder and president of Global DARE Foundation. And I'll be your host today. My co-host is Susan Karanoff, who is our secretary at Global DARE Foundation. We're honored to have Professor Ronald Wanders and Dr. Sacha Ferdinandussa from the University Medical Center in Amsterdam as our presenters today. All right, having a little bit of technical difficulty. Oh, here we go. For some reason, I was having difficulty moving the slides here. So here's our hosts and presenters. So now let's move on to the agenda. So I'll be providing a few housekeeping details to get us kicked off and then review our Global DARE Foundation's mission. I'll then hand it over to Ronald and Sacha to present on the science behind Refsum disease. Following their presentation, Susan will begin the question and answer portion of the webinar. Now on to the housekeeping details. So all participants are in listen-only mode. There will be a few ways that you'll be able to ask questions during the webinar. Participants following on Zoom can type their questions in the Q&A box at any time during the presentation, or by raising their hand by pressing Alt plus the Y button on their keyboard, or click the icon on the toolbar in Zoom. Participants joining by phone can press *9 on their phones to raise their hands as well. After the presentation, Susan will be moderating the questions. She will start with the questions in the Q&A box in Zoom, then the dial-in participants, and then to the online participants. I think something happened to my video there. So in today's session will be recorded for later viewing on Global DARE Foundation's website. All right, I think I'm having a little technical difficulties with my video, but we'll get going anyway. So Global DARE Foundation was established in October 2019. DARE stands for Defeat Adult Refsum Everywhere. Our mission is to promote worldwide awareness and better quality of life for all who are diagnosed with adult Refsum disease. Our goal is to support research, education initiatives, awareness campaigns, and advocacy. Driving research is at the center of what we do because we dare to believe in a cure for adult Refsum disease. So now I'll be handing the presentation over to Ronald Wanders, who will give us, we'll start our presentation around the science behind Refsum disease. So Ronald, I think you just need to unmute yourself, and then I'm going to give you control of the screen, and you can go ahead and be able to work through your presentation.
Okay. First of all, it's an honor to give this presentation to all of you. Some words about myself. I'm a biochemist by training, actually, and after my PhD, which I did a long time ago, I always wanted to work in a hospital setting. So when I got the opportunity to come and work here in the AMC, where I'm still at the present time, I took the opportunity. So I basically started from scratch as a postdoc and all the way up to, become head of the laboratory. And as most of you know, we have a long-standing interest in peroxisomal diseases, including Refsum disease, from the very early moments on. It all started with patients presented here to the Department of Pediatrics, and that got us to where we are now, actually. And we know lots about the peroxisomal disorders. And we did a lot of work on Refsum disease in the early days, but because of funding reasons, we had to stop it. And Sacha will speak about the work we did, for instance, on the mouse model, and which we would like to use again for, to get behind new therapeutic options for Refsum disease. But I come to that at the last part of my presentation. That's as an introduction. Do I advance myself now, Christy? Do it? Or you do it?
Christy, you're on mute.
Sorry, you can go ahead, Ronald, and flip through your slides now.
It doesn't work.
It doesn't work. Christy, you do it.
No. Okay. Let's see. I should have. Let's see. Okay, I can flip through for you. Let me see if I can try to give you control again and see if it allows you to have it. You want to see if it'll? Yeah, there you go. Just put it back in presentation mode. Oops, sorry. There you go.
Yeah. I started off with a very basic scheme, actually. I called it metabolism in a nutshell. It's just simply to tell everybody that we have three types of food which we take in on a daily basis. That's carbohydrates at the left part of the slide, which will be converted into simple sugars like glucose. And a part of it gets oxidized in cells because we need energy to think, to move, etc. And in part, the simple sugars like glucose, they're also converted into glycogen as a storage form of sugar in our body. And we also use them to synthesize more complex sugars in our body for other purposes. In the middle, you find the proteins, which consist of amino acids. And they are taken up in our intestine and incorporated into our system via the blood system. And ultimately, amino acids can be degraded to acetyl-CoA. That's a kind of central element in metabolism. And if that gets broken down, you'll have ATP and you'll have your energy which you need. And also amino acids are partly broken down and partly also used, for protein synthesis in our own body. And finally, for the purpose of today, that's the most relevant part. That's where fat comes in, and fat consists of fatty acids, and they are also degraded in our body and ultimately give us ATP and energy as well. And in part, fatty acids are required for de novo synthesis of various lipids in our body. That's as a starting point. Second one, Christy, second slide. Yeah, it works. So phytanic acid, which is the topic of today, that's a typical fatty acid. So we're only dealing here with the right part of the figure. So there's in Refsum disease, there's no problem whatsoever in terms of carbohydrate metabolism nor protein metabolism. It only has to do with fat metabolism. Well, a few words about fatty acids. The general structure is shown on top. There's this structure which I indicate in yellow. That's the end of any fatty acid, and next to that there's this side chain, and that can have various forms because fatty acids can occur in all kinds of different forms. They can be short, like the one I showed here, but they can have a medium chain length and they can be very long. And phytanic acid is such a long chain fatty acids. I'll come to that in a minute. Fatty acids can be saturated, and then they have no double bonds, or unsaturated. And unsaturated fatty acids, for instance, are linoleic acid and oleic acid, which you might know from the food store, basically. And then again, fatty acids can have branches, and phytanic acid is such a branch chain fatty acid. And the structure of phytanic acid is shown at the bottom. You recognize this at the end of the molecule, which makes it a fatty acid. And then if you look at the rest of the molecule, you see all these branches. This is one branch, this is a second branch, and this is a third branch, and this is a fourth branch. And these branches, they basically make metabolism different. I'll come to that in a minute. So most fatty acids are broken down in mitochondria in our cells, again to make energy. And the way that happens is by means of cleavage of the molecule. So what I indicated here with a pair of scissors. So if this is palmitic acid, which very much resembles phytanic acid actually, you get cleavage here. And then this part of the molecule is released as acetyl-CoA, and this then can be degraded in our system, in our human body, ultimately to give CO2, which we expire, and water, which we, get rid of by other means. And then what happens is that the fatty acid is now chain shortened, and then the same thing happens again, ultimately until the fatty acid is completely degraded and cut into small pieces and turned into energy, basically. This is the structure of palmitic acid, and underneath I put the structure of phytanic acid. And if you look at it, it's strikingly comparable. The only difference basically is that you have these branches again, the 1, 2, 3, 4 branches, and that makes metabolism different. And I'll come to that in a minute. So phytanic acid, because of these branches, cannot be broken down by beta oxidation, like palmitic acid can, and it requires a different system to be broken down, and that we call alpha oxidation. So Refsum disease is basically due to a defect not in the beta oxidation that runs perfectly well. Refsum patients have no problem whatsoever to deal with and degrade palmitic acid or oleic acid or whatever fatty acid, but you do have a problem in getting rid of phytanic acid. In the early days, we figured out actually the precise mechanism by which phytanic acid is degraded in our human body. It's a bit of a complicated system, basically consisting of 5 different enzymes, 5 different steps, which have to work, otherwise you have a problem and something piles up. So if you have a defect, for instance, at this level, at number 2, which is the defect in Refsum disease, I'll come to that in a minute. What happens is that obviously the products upstream of this lock, because that's what it is, if this is closed down, then you'll have accumulation of everything upstream, and you have a shortage of all the molecules running downstream. So the end result of this mechanism, which we call alpha oxidation, is that this molecule, phytanic acid, is shortened by 1 carbon atom. And so what you have then in the end is this molecule here, phytanic acid. And the funny thing is that also has 4 branches, just like phytanic acid, but because it's now 1 carbon atom shorter, this one can be beta oxidized. So that runs off smoothly, no problem whatsoever. The only problem is here in getting rid of this end part of the molecule. And that happens once more via this set of 5 different reactions. In Refsum disease, we published that a long time ago. It was one of our first PhD students, Herbert Johnson, on the topic. And he set up an enzyme assay to measure actually this enzyme. As you can see here, it's very deficient. If this is the control, we measure it in the control fibroblast, it's 100%. And in Refsum patients, it's never 0 actually. I'll come back to that at the end. But there's always a few percent left. Yeah. So next question then is, if this is the metabolism of phytanic acid, I hope you understand a bit. Then the next thing is the sources of phytanic acid. Most fatty acids in our body, they come from outside sources, from food obviously, but they're also synthesized endogenously. For phytanic acid, life is simple because it only gets in from exogenous sources. We cannot synthesize it ourselves in our body. So, about the sources of phytanic acid, it's either phytanic acid itself, which comes in from the diet as a free phytanic acid, so not bound to anything else, but it's also clear that phytanic acid comes in several bound forms. And then in our body, and to ultimately be degraded. And these free fatty acid and bound fatty acid is present in all kinds of phytanic acid foodstuffs, butter, meat, certain fishes, etc. Again, I'll come back to that in a minute. And there are also these phytanic acid precursors, especially Phytol. And something new actually here, that's these phytyl fatty acids. It sounds very complicated, but it's not. I'll tell you in a minute. So, but Phytol, the structure of Phytol, if you compare that to phytanic acid, you can see easily that it's very comparable. And actually what happens is that Phytol in a number of different steps is converted into phytanic acid. Work by another student, Daan van der Brink, led to the resolution of the pathway which handles Phytol and converts it into phytanic acid. So this is the structure of Phytol again, and it's important to read the text. Phytol is a direct product of chlorophyll, because Phytol is an essential element of the chlorophyll molecule as present in plants. And it's released from chlorophyll by ruminants, but not by humans. So humans can eat chlorophyll in the form of spinach or whatever, and that will not lead to any burden of Phytol or phytanic acid. That's good to know. It only happens in some animal species, especially ruminants. And they are able to release the Phytol from the chlorophyll molecule, and then the Phytol is either stays like that, or it's converted into phytanic acid, either as such or coupled to other metabolites. And at the bottom of the slide, it's in a simple scheme. So chlorophyll will be converted into Phytol in animal species, including ruminants. And then in our human body, it can go two ways. It can be converted into phytanic acid or into phytyl esters. I said in human body, it's not true. In the ruminant body, it's converted into phytanic acid, but also into phytyl esters. I come back to that in a minute. These fatty acids, actually there was work published a few years ago actually. It was quite exciting because that was a new thing which we did not know so much about in terms of Refsum disease. Work from a German group, and they clearly showed the presence of these phytyl fatty acids. That's nothing more than a Phytol molecule on the one end, and that on the other hand, there's fatty acids, and they're bound together. So they're basically one composed out of two different molecules. And this is the structure, but that's maybe a bit complicated. But the essential thing for today is that you have free Phytol, which is in foodstuffs, but there's also this bound form of Phytol. And then these Germans showed, I think that's in my next slide, they showed here that these phytyl esters are present in all kinds of different fruits, like in cucumber, only a little bit, in red grapes, in green olives, in carrots, but especially in rocket salad, and also in some bell peppers. If you look here at the amounts, it's quite appreciable. And what you also did, because you never can tell beforehand if such a molecule like this Phytol bound to a fatty acid, is that really entering our human body, yes or no? And that's an essential aspect which is always difficult to resolve. But they did a very simple experiment, these Germans. What they did was they took saliva, because if food enters our body, it first gets into your mouth and it meets your saliva, then it gets into your stomach, and then you have your gastric juice there, and ultimately it enters our intestine, and we have this intestinal juice. So what they did, the Germans, they synthesized actually this phytyl fatty acid ester, that is Phytol bound to palmitic acid in this case, and they wondered and asked the question, is this degraded by any of these three different body fluids, because that's what they are. And they showed that saliva is not able to release Phytol from this molecule, so no problem. Gastric juice, also nothing is cleaved. But when they added the intestinal juice, then they got release of the Phytol. So telling us that it's a real risk factor for Refsum disease. Actually, that's in their title. As you can see it there, it's a risk for patients suffering from Refsum disease. Reason being that in our intestine, we have these enzymes sitting there, lipase, here it is, and that's able to cleave the molecule so that you'll have Phytol released, and that's it then incorporated, taken up in your intestine, gets into your system, and then you have potentially a problem, because then that generates Phytanic acid as well. Is that clear? Yeah, I think it's clear. Conclusion. And we came to that conclusion actually in the last weeks or so, when we discussed about this item with Susan Kuranov and Ann Moser and several other people, the Rada from London. And we concluded that it would be wise to check, and we will do that, as many food components as possible. Not only check Phytanic acid and also bound Phytanic acid, but also free Phytol and the Phytol which is bound, as in the case of the phytyl fatty acids. We agreed on that, I think it was 2 weeks ago, Susan, that we will do so. And Moser will take the lead here. So far, so good. For the last few minutes, what about the future? So what we agreed on with the consortium, with the DARE consortium, is to reevaluate the contents of Phytanic acid and Phytol, both free and estrophyte, in all kinds of food components. We will do that under the leadership of Susan and Christy, of course. And then the other thing, apart from the dietary aspect, is of course the future in terms of are there any methods to circumvent or correct the enzyme defect in Refsum disease, because that's our ultimate dream. In the future, in one of the future webinars, we will discuss gene therapy and also enzyme replacement therapy, which is very successful, for instance, in other inborn errors of metabolism, especially the lysosomal storage diseases. But for now, there are also two other options. Number 5 is upregulation of residual activity, because I showed you that in Refsum patients, usually there's a few percent residual enzyme activity. So, you know, if we would know how to induce this activity by one way or another, and we have experience with other defects which we work on in the lab, that would be great. And that's something we would love to do in the future as well, provided we have funding, obviously. And then the other thing is an alternative route of Phytanic acid breakdown, which was already hinted to by Gisbiki in the last webinar, but it's good if I tell a few words about that as well. So here is Phytanic acid again. It's impossible to degrade Phytanic acid via the regular way, via alpha oxidation. So many years ago, we already thought, and Tony in London as well, actually, is there a way to break down the molecule from the other end? Because, you know, if you would chop up the molecule from this end, then you will basically have the same end result, and that is that Phytanic acid is degraded. And we worked on that, another PhD student in the lab actually, and we figured out that indeed Phytanic acid already under normal conditions is almost oxidized. So it's possible to degrade Phytanic acid from the other end. And then you don't have this problem introduced by this methyl group, because from this end, it runs independent from the alpha oxidation. It's only beta oxidation. And that's the good thing of eating up the molecule from the other end. And we figured out the enzymology. So there are a number of enzymes which we call cytochrome P450 enzymes, which take the first step, and then an alcohol dehydrogenase comes in and an aldehyde dehydrogenase. And then you have the molecule ready for beta oxidation from the other end, as I show here. And the end product of this pathway is 3-methyl adipic acid. And the good thing is that Tony already did some work on it in his patients. And that's shown here. Is this the right figure? I'm not sure. I made a mistake, probably. A bit stupid. Anyway, what Tony showed is that the Refsum disease patients, they have this omega oxidation pathway, you know, eating up the Phytanic acid molecule from the other end, operational. And he showed that because 3-methyl adipic acid, it's in the title here, is end product of omega oxidation, and that correlates pretty well. I picked the wrong figure, stupid enough. But anyway, it gave a more or less linear relation with Phytanic acid on the y-axis and 3-methyl adipic acid at the other axis. It runs like this, if you follow the red line a bit. So this leads then to the idea that if we would know a way of or means to upregulate omega oxidation, then we would be able to induce the capacity in Refsum patients to catabolize Phytanic acid. And that's shown here. And that's actually where we left our studies, and then realized that we needed a mouse model to figure this out any further. And Sacha is going to speak about that in a minute. She did the work in those days as a PhD student, and then later continued as a postdoc. And now she's staff member in the laboratory here and a researcher. And both of us, we still share this fascination for Refsum disease. But the idea then was that we needed a mouse model to see whether the things which will work in vitro, in cell models, you know, is that really working out in the mouse model as well. So we generated the mouse model, Sacha will speak about it. But the ultimate idea there was, can we then induce this alternative route by manipulating, by inducing, by upregulating these enzymes, you know, which catalyze the first step in the omega oxidation pathway. And that's basically where we left it, or had to leave it actually, because of funding issues. Exciting still. And that's then the end, as far as I'm concerned. We really have an Amsterdam Refsum team. It was a bit quiet for a while, but thanks to Christy, we all got motivated again. And there's the lab team here with Sacha and Fred Voss, who's actually running the metabolomics department in our lab. They're married to one another, Fred and Sacha, so that makes a happy combination here in the lab. And there's Hans, who's running the molecular biology unit and looks at Refsum disease from that point of view, and that's me. I have a background more in metabolism and cell biology. Then we have the clinicians in-house, Mark Engelen and Carl Hollack, and they see our Refsum patients actually, especially Carla. Corrie Timmer, she's our dietitian. She's taking care of the Refsum patients we have on board in the hospital, not so many actually. And then we have Clara van Karnebeek and especially Arthur Bergen, who's an expert in eye research actually. And we got him on board the Amsterdam Refsum team as well, because he has experience with retinitis pigmentosa and all kinds of stuff regarding the eye. And we are happy to have him on board. And also one of the future project we have in mind is strictly focused on the eye and Refsum disease. That's me. Well, it was nice. Thank you. Now Sacha will join in.
Yes. Thank you, Ronald, for this introduction, and thank you all for giving me the opportunity to tell you something about the work I did. Indeed, I'm a laboratory specialist working now in diagnostics and research. But I remember well when I started here over 20 years ago as a student on an internship on Refsum disease, because that was right at the time when here in the lab the molecular basis for Refsum disease was discovered. So that was a very exciting time, and it really got my interest. And I still work here after all this time under supervision of Ronald. So I'd like to tell you something about the work we did on the mouse model for Refsum disease. I did this work with Pedro Brites, who is now in Portugal. And well, we generated the mouse model by disrupting part of the phytonyl-CoA hydroxylase gene. And you do this by making a deletion of part of the gene in embryonic stem cells of mice. So, and as a result, you do not get messenger RNA nor protein expression, and also the enzyme activity is gone. Now you see in this picture here, ++, that stands for wild type or control mice. And as you can see, here is expression of the phytonyl-CoA hydroxylase because this mouse has 2 wild type normal alleles with the phytonyl-CoA hydroxylase gene. The ± stands for heterozygote, so you can compare it to a carrier. And the ± has 1 wild type allele and 1 allele where the hydroxylase gene is deleted. But because of the wild type allele, you still have expression of the protein and enzyme activity. And the -- is a homozygote mouse, so it has 2 alleles with a deleted phytonyl-CoA hydroxylase gene. And in the presentation, I will call these the knockout mice. And as you can see, there is no expression of the phytonyl-CoA hydroxylase anymore. Now, these knockout mice are viable and fertile. And well, nowadays, it's relatively easy to generate a knockout mouse model. It was not that easy when we started this project, I think already over 15 years ago, but we ended up with a knockout mouse. But then the most important question is it a good model for human disease? And that's not evident because of the many differences between mice and men. So that was the thing we had to find out, and I will tell you about the studies we did to investigate this. Well, the first thing we realized is that there is no phytanic acid in normal mouse chow. So we have to feed the animals with a phytol supplemented diet to generate an even potentially good model. So we used these wild type control mice, we used the knockout mice, both males and females, and we supplemented them with a control diet, 0.1% Phytol diet and a 0.25% Phytol diet. And we did that for 6 weeks, and the highest diet, the highest Phytol diet, only 3 weeks, because the knockout animals did not really handle the Phytol diet very well. So it's a relatively short period of diet. And then we wanted to look at how what the phenotype of these animals is. So we did certain tests at the start of the experiment and at the end of the experiment. We did a Sherpa analysis, which is a systematic and objective protocol for phenotype analysis. I'll come back to that later, and a catwalk analysis. And we did that at the start and at the end. And at the end, we sacrificed the animals, we isolated the tissues to do biochemical measurements and to look at the tissues, so do histology. Now, the first things we noticed was that the knockout animals on the Phytol diet, here you see such a mouse, weighed much less. So the animals, the knockout animals already weighed less at the beginning, and they lost a lot of weight on the Phytol diet when you compare it to a control mouse. Now, this is really something that is different between mice and men, because when we sacrificed the animals, we saw that almost all the white fat is gone. But this is a mechanism which is really specific for the mice and not for men. So I will not go into that in detail. We measured, of course, phytanic acid levels. We did it in plasma, and you can see the results here in this inlay. Here you see the control diet, Phytol diet, the 0.1% and the 0.25%. You see the knockout animals, that they accumulate phytanic acid in plasma up to 1 millimolar. So, and also in the tissues, you see here in the knockout animals, on the Phytol diet, a huge accumulation of phytanic acid, and we looked in liver, kidney, cerebellum, so the brain, and testis. So we could conclude that biochemically, at least, it's a good model for Refsum disease because it has the biochemical hallmark, the accumulation of phytanic acid. So then the phenotype. So like I said, the Sherpa is a systematic and objective protocol for phenotype analysis, and you do 24 little tests before and after the diet in a standardized way, and you score the way the animals behave. So first you observe the animals, you observe how they walk, what the pattern is, and then you do some short manipulations like picking them up at the tail. If you do that to a mouse, it will automatically turn around and try to climb up to your hand. But of course it needs the coordination and the strength to do so, and if it doesn't have that, you can score it as negative. Now I will show you a short video of I think 2 tests which we did. Here you have the mouse on a grid, and if you turn it, flip it over, the mouse will turn around and climb up, and then you can score whether it can do it or whether it has pulse slips while doing it. And here you see the animal in a cylinder, and again, if you turn the cylinder, a mouse will turn around, and again you can score whether the mouse is able to do this. Now, we saw that the knockout animals on the Phytol diet scored abnormally on some tests. It showed an increased pulse slip, an absent trunk curl, and abnormal movement. So we found signs of abnormal neuromuscular function in the knockout animals after the Phytol diet. So to analyze this in more detail, we used the catwalk. Now, what's a catwalk? A catwalk is a glass plate, and the animal has to walk over the glass plate. It has to do 3 fluent runs, and you have a closed box with a light source, and the light shines along the glass plate, and wherever the mouse puts its paw down, the light is reflected and the camera stores this information. Now here you look from beneath, and here you see the mouse, and you see where it puts down its paw. But to be able to calculate anything, you have to label by hand each paw print. So every time he puts his paw down, you have to label it and tell the computer, this is the left forepaw, this is the right forepaw. And if you do that, then the computer can calculate all sorts of parameters like the step sequence or the base of support of the paw print area. Here you see how we do that. You see the mice walking along the glass plate, and that we have labeled each footprint. And then you can make a printout, for example, of 1 run of an animal. These are only the hind paws. Here you see a printout of wild type animal, and here the knockout animals, males, females. And if you compare, if you give it a control diet, you see no difference in the paw prints between the wild type and the knockout animals. On the Phytol diet, you do not see any effect on the paw prints from on the wild type animals, but in the knockout animals you see a big difference. So you see a decreased paw print area, for example, so the mouse really has abnormal gait. So after the Phytol diet, the knockout animals showed a decreased base of support for the hind paws. They showed abnormal and decreased paw print area and an abnormal wind off. So the knockout animals show gait abnormalities, and they point to a neuropathy. And that's why we did motor nerve conduction velocity studies, where you stimulate the sciatic nerve, so a nerve in the leg, and then you look at the output. And here we saw that the knockout animals on the Phytol diet have a decreased motor nerve conduction velocity. And here you also see that the amplitude of the output is decreased. We also looked at myelination by histology, and this was normal. Now we also looked in the central nervous system. We looked at the cerebellum, which is part of the hindbrain, which you see in red here. And the cerebellum is very important for movement and motor learning, and we looked especially at the Purkinje cells. Here you see a very nice picture of a staining of Purkinje cells. They are the neurons, this is the cell body, and here are the branches, the dendrites. And we did this staining also in our animals. Here you see the wild type animals, and here below the knockout animals. And this is the control diet. There you see no difference between the wild types and the knockouts. And when you start giving the Phytol diet, you see no effect in the wild type animals, but in the knockout animals on the low Phytol diet, you see focal loss of Purkinje cells, and you see actually quite big loss of Purkinje cells on the high Phytol diet. And we've also quantified this, and you see that here in this bar graph that the number of Purkinje cells is much reduced in the knockout animals on the Phytol diet. So to conclude, the knockout animals on Phytol, they accumulate Phytanic acid in plasma and tissues, so biochemically it's a good model. They develop a neuropathy and they show cerebellar ataxia accompanied by loss of Purkinje cells in the cerebellum. So we concluded that potentially it's a good model to study therapeutic strategies, because if you have a therapy, you can test whether it can lower the Phytanic acid levels, and you have a readout, for example, the gait abnormalities. You can see if they improve after the therapy. So that's actually what I wanted to tell you. So thank you.
Thank you very much, Sacha and Ron. Very interesting presentations. Christy, are you on mute, possibly?
Yes, I'm on mute. Sorry. I think at the moment we don't have any questions in the Q&A box at this point in time. Does anyone in the audience have any questions for Sacha or Ronald?
Well, I'm glad that everything was clear.
Oh, I think we have a question coming in.
Yeah, so in the mouse model, was there a difference in the effect on male and female mouse? And would a different model, a different animal be better?
No, we did not see many differences between males and females. So in that respect, it's a good model. But we know, as you've probably noticed, I did not tell anything about the eye. We did not study it because we did not use the right background, because when you make these mice, they come in a certain background, and it was partly Swiss mice, and they're not practical for eye studies. So we started backcrossing to a different background, so it should be possible. But actually for the eye, a rat, I've been told that the rat model would be better to look at a retinopathy. So, but like I said, for example, testing out gene therapy, this is good enough.
Okay, thank you. Another question. So firstly, a great talk. Thank you very much. And where are the slightly increased Phytanic acid levels in the knockout mice on regular chow, phytol-free diet coming from?
Well, it's probably that there is still some Phytanic acid or Phytol in the normal mouse chow. So that it will be diet derived, definitely. But it was just not enough to induce a phenotype. Maybe actually, because we like I said, we looked short term. Of course, mice, they live about 2 years. So what you actually should or could do is to study them on a regular diet, and then also see after 2 years if they without a Phytol supplemented diet, to see if they develop any symptoms, you know, to see if the small amount in a prolonged period will also cause a phenotype. But we didn't do this yet.
Mm-hmm. Okay, there was a question that came in as well prior to the meeting. So I've noticed that in our Facebook chats with other Refsum people that there are different onsets of severe symptoms at earlier ages than others, noticeably during teens. So what is the likelihood of what the mother eats during the pregnancy affecting the Refsum baby she's carrying?
Yeah. What do you think, Ronald? I think, you know, the mother has normal metabolism. Yeah. So I do not think it will influence this much. I think the difference will be after birth. Yeah. Because of there will be genetic differences between patients. So I think that will be the biggest determinant, and not in utero.
Yeah. Although I remember from the old literature, but Tony knows better than I know, that there are some bone abnormalities which are already there from the very first moment on, actually. But I doubt whether that has anything to do with phytanic acid. This phytanyl-CoA hydroxylase must do more than only, you know, degrading phytanic acid. That's always been our hypothesis, but we never got a hand around it. But that's one of the things which I remember from the discussions we had when we had a European project on Refsum disease with Tony and Dr. Gibbert from London. Maybe it's good to ask Tony about that, Christy and Suzanne.
Mm-hmm. Yeah, we could follow up on that with Tony.
Yeah.
Yeah.
But I think what Sacha is saying, that's how it is. It's probably all determined after birth, you know, when phytanic acid starts to pile up.
Right.
So Susan, I have a question. This is Christy. So I know when on one of the slides, Sacha, you showed the accumulation of the phytanic acid being in, you know, highest in the liver and then the kidneys and then so on. Was there any ever testing of whether, because I know several Refsum patients have joint issues, you know, and whether there's had been any accumulation of phytanic acid in muscles?
I will have to check. I think we did it, but I will check. We certainly did all different kind of nerve tissue, and there was clear accumulation. I think I will check for you and let you know, but there must be. I mean, it's actually, we did so many different tissues, and there was accumulation in all tissues.
Mm. Yeah, just curious as to whether, especially as I said, we've seen several patients with joint issues, and it's not necessarily something that's called out in as Refsum as being an issue, except for from some of the bone abnormalities, plate issues, things like that, and whether there's anything more to it than just that, you know, and whether, depending upon someone's activity level or depending upon how they present, whether those joint issues could be related to phytanic acid, and because I believe peroxisomes exist in the muscles as well, right? And if you know fatty acid makes it to the muscles, there could be accumulation there, and then release from there. I would assume too, maybe that could potentially deteriorate those muscles. I'm not sure, you know, I'm not the expert, so, but just, you know, theorizing myself. So,
So, Sacha, as a follow-up question on that topic, if phytanic acid is accumulating in the kidneys, is that going to eventually impact kidney function?
Yeah, it could. I don't know if it was ever observed that I would prefer to ask to a clinician. We looked at histology in the kidney, and that was normal. That was not affected in these animals.
The histology was normal.
Yes.
Yeah.
But like I said, that it was a short-term experiment of only a few weeks. So, but there we did not find anything.
Right. Okay. Because when you read in the literature, kidney issues are mentioned, although they're not as frequent as some of the other symptoms. But cases of kidney issues have been reported. Yeah,
but it could, of course, on long term. Yeah.
All right, Susan, I think we have one question from Judy. So I'm going to open up the line for her to ask that question.
Okay.
All right, Judy, I think you just need to unmute yourself.
Unmute yourself.
I think.
Let me unmute her.
I can't. I can ask to unmute, but oh, there we go.
Are you okay to ask?
Yes, you're good.
Very good. Well, most Refsum patients exhibit a common theme of losing eyesight and so on, RP. However, the great majority of them are not exactly the same in terms of the areas of weakness they exhibit. And nearly almost everyone has a different form of weaknesses as a result of Refsum disease. How do we explain that?
Yeah, that's for me difficult to answer, especially since I'm not a clinician, so I cannot. And do you have anything to?
No, why the patients are so different? Is that the question?
Yeah,
Well,
I understand that.
The differences in hearing, for example, in smell, and in gait.
Yeah, there will be, of course, one obvious thing will be residual activity.
Yeah.
So that will be different, and there will probably, because we know a lot of examples of genetic modifiers in inherited disorders. So it's a very well-known common phenomenon, and there are even disorders like X-linked adrenoleukodystrophy where the differences are even much more striking, where within one family with the same mutation you get a different phenotype, completely different phenotype. So we know that there are modifiers, and so there will be modifiers also in Refsum disease. Next to the fact that if you have different mutations, you get different residual activities. So this will also depend. And then because Refsum disease is also based on how much Phytol you get via your diet. So there are so many factors which modify this. So it will be very difficult to pinpoint one thing, but there will be a lot of things, an interplay in metabolism, I think. Yeah, omega oxidation, like Ron says, could be of influence, of course. This could be more working harder in one patient than in the other. So.
Thanks, Summer, for the question. This is Christy. I have another question. I don't want to dominate the questions, but if there's none, I'm going to ask some. So, did you ever do, Sacha, any testing on the carrier mice?
Well, only briefly, because they seemed completely normal. So we did not do extensive studies. So only in the beginning. But like I showed you, there is actually normal expression of the phytonyl-CoA hydroxylase. So we did not really continue with that.
Okay. And I know I was recently researching Phytanic acid and its role in other things like prostate cancer and lymphoma. Did you guys ever take a look at some of that as part of information around Phytanic acid? Or I know it was years ago, I think they, back in the 2012, 2010, they did some research on that.
We did, especially, it is shown indeed that the whole branch chain fatty acid metabolism, there is an association with prostate cancer, and we actually were more involved with the subsequent enzyme, the racemase, the alpha-methyl acyl-CoA racemase, and there the expression is very much linked to prostate cancer, and also even more downstream the bifunctional protein. But I don't think we did it for the phytanyl-CoA hydroxylase.
No, we did not.
But well, the whole pathway is certainly linked to that.
Is there any connection between the PHYH and the AMACR? Is it like any connection between those two? You know,
well, they're in the same pathway. So so yes, and for that reason, it's the disease is similar on certain aspects.
Okay.
Yes, well, they also accumulate Phytanic acid, these patients. Yeah. Also, as a consequence, you have the block further down in the pathway, but because of that, you also accumulate Phytanic acid if you have a racemase deficiency.
Yeah.
Okay, we have a further question in the Q&A. I think we still got time for one or two more. So again, thanks for an excellent presentation. Now, this is the million-dollar question. So what is the next step in terms of research following the fascinating mouse model work? And a follow-up question, what timeline do the speakers believe it would be expected to make a breakthrough with Refsum? And is there sufficient funding to progress the work?
Yes. Well, I think the most logical step would be the liver gene therapy, because I'm not an expert, so it's very good that there will be a next session on this topic with experts. But there is a mouse model, and there is expertise in the scientific community to do the gene therapy, and if you restore the phytanyl-CoA hydroxylase activity in the liver, potentially that should be for many symptoms enough. The question is, of course, if it will be enough to restore the eye problems. So that will be, I think, a different, an issue. Also to test that, we wonder whether we need the rat model. So I think, that is the obvious way to go. Enzyme replacement therapy is less obvious because you would need to get the enzyme in the peroxisome. And for the lysosomal storage diseases, there is a good mechanism to do that, to get the enzyme to the lysosome, but for the peroxisome, it's not easy. So I don't think that's very feasible. And funding, well, I do think it's a problem. There's not much possibilities to get funding for these studies, even though I think it would be a great breakthrough for the entire peroxisomal field, because I think the gene therapy on the phytanyl-CoA hydroxylase gene is the most the best achievable, or how do you call it? For example, when you compare to the peroxisome biogenesis disorders, it will be much harder to restore those. So it would be great if we could do it and could get the funding to do it.
And we were working on that actually together with Joe Hazia. The idea is to generate an NIH project, And also within Europe, you have this e-rare program, which we are trying to work on, you know, especially with Bart Leroy, also from Ghent. He's also an expert in the field of Refsum disease, and the people from London. So from these two angles, we try to generate a project, because that's obviously very much needed, because there are good ideas now, and liver gene therapy is very feasible nowadays. So I think we have a good chance of getting somewhere if we get the funding properly done. And in the meantime, together with Ann Moser, we will work on, you know, the content of Phytol and Phytanic Acid and all the different foodstuffs. That's for the time being also very important, but that's not so costly. So we'll do that anyway. I think if we would get that to work, we are in business with respect to Refsum.
Yeah, just to add a little bit on the funding side, you know, obviously we've got a really great team in our medical and scientific advisory board, as Ron and Sacha were just mentioning, a great group of experts, and we are driving forward some grant research. Another key piece here is finding more patients, right? So we, you know, to make it attractive to investors to move forward a gene therapy clinical trial, it'll be really important to find more patients. To, you know, it's a profit business out there as well, and so we have to realize that, and so we have to make it attractive for them too. So we'll be doing a lot of work in the foundation in parallel to some of the initial proof of concept work, maybe within the gene therapy space, to really find more patients to make it attractive for an investor to move to clinical trials. So lots of things in parallel. It's not going to happen overnight. We've got to have some staying power and push forward on it, and we'll get there eventually. And the more support we can have from patients and their families, and it'll make a difference, right? So I think we might have one more question.
Yeah, Chris, it was just a question whether we're working with the team at St. Thomas's. Yes. And I actually just typed an answer to say that, well, some of the members from the St. Thomas team are actually members on our medical and scientific advisory board. So it really is a, you know, a cross institutional effort that we're trying to push forward here.
Yeah.
Yeah, I recently did a presentation, which is on our website as well, that covers off on our medical and scientific advisory board and some of their objectives. And so if you're interested in learning more about what we're doing at Global DARE Foundation to help, you know, move forward our mission, I would recommend watching that video or reaching out to myself or Susan or anyone here at Global DARE, and we can tell you what we're doing to help move that forward. But we've got a great, I'm very proud of the medical and scientific advisory board that we have established. They, you know, obviously bring a lot of great experience to the table, and I think, you know, with some momentum, new momentum and excitement around it, we can hopefully make some great progress.
I agree, Christy. Well done.
Yeah, great. Well, I'll let you finish off, Susan.
Right. Okay. Do you want to bring up the last slide, Christy? Just to briefly remind everybody that we do have more webinars coming up. So today concluded the second webinar, and later this month we have the Refsum diet overview and discussion with the two dietitians from the St. Thomas Clinics, Eleanor Baldwin and Sarah Furman. And then in August, as Sacha alluded to, we have a presentation on gene therapy given by Ryan Butler from Southwestern University in Texas. So if you haven't already signed up for these, you can do so on the DARE Foundation website.
Yeah, great. It was nice. It was real nice.
Well, thank you everyone for joining today, and that concludes today's webinar. Thank you.
Okay. Bye, everybody.
Bye.
Thank you very much, everybody. Bye-bye.
Bye.